So today we're talking about continuing with blood, and we're going to get into hemopoiesis in some detail. But I'm starting this out just talking about the biotech companies and what is in their pipeline. So saying that a pipeline is the development of products, all the products that are, they might not be offered yet, but they're being developed, that's considered their pipeline. It's particularly important for pharmaceutical and biotech companies. And the reason for that is that it takes a long time for stuff to come through the pipeline. So if you want to keep your investors interested and you want to keep the doctors and hospitals paying attention to your product line, you want to advertise that a little bit up front. And then in addition, you have to do clinical trials and everything. And it's a big gamble for the pharmaceutical biotech companies to run clinical trials because it costs a lot of money. Have you heard about that? No, not so much. It's good stuff to know about though, because as I was saying, the biotech companies are big in the Bay Area, and they merge actually with pharmaceutical companies. They're constantly being bought out by pharmaceutical companies, or at least deals are being made between biotech and pharmaceutical companies. So there's quite a bit to know. And a lot of these products do focus on blood. So that's the reason why I'm bringing that up now. Not all of them, but a healthy group of them. So cancer is obviously very dominant, and that's because it's a common disease, and a lot of people get cancers, and so having an anti-cancer drug is a big deal. Genentech's anti-cancer drug has done extremely well, and it's basically a monoclonal antibody against VEGF. Let's see, not on this one. As far as what kind of drug it is, let's just look at Amgen's pipeline for a second. Did you guys ever hear of Amgen? No? You've heard of Genentech though? Yeah. So Genentech are the local guys in South San Francisco, and I worked there for a while. Amgen had presence here as well, but they're located in Thousand Oaks, just north of LA. So this describes their pipeline. So I just put in the Google search Amgen pipeline. And here what you see is that they have different, what they call modalities, different types of molecules, classes of molecules that are therapeutics. I think if you're interested in medicine, being interested in the drugs that you're going to be prescribing is a big deal. That's basically a big part of your training, is they're going to teach you, what sorts of therapies are possible, what kind of medicines are available, what's the right medicine to prescribe with a certain diagnosis. So the connection between these therapeutic molecules or drugs and medicine is really intimate. There's a big connection there. And so, that's why I'm trying to introduce some of these things into the class, because it is important. And there's a lot of the questions you'll be asked have something to do with, trying to understand what types of therapies are out there. So I'm showing this one, Amgen's pipeline at the moment, and every line here has a different type of molecule, and that's another potential product that they're coming up with. And some of these are out in the market, and some of them are not. What does it take to get a drug out on the market? It takes an investment of millions of dollars, like typically hundreds of millions of dollars, and typically more than a decade of research and clinical trials. It's really expensive, so they're really betting a lot on the success of a particular drug. And then once they start to release it, if some of the patients have a side effect, they could potentially sue the drug company, and then the drug company would be under pressure to withdraw the drug from the market. You guys have heard of that sort of thing happening, right? So it's a really risky process. Not so simple. But anyway, there's different types of molecules, which I think is kind of cool. It used to be that drugs were just small molecules. You know, a lot of times derivatives of plants, things like aspirin, you know, is a derivative of plant. A lot of things that didn't really understand what was going on. They would isolate some complex molecule and find that it had some efficacy, and then you know, you sell that and you hope for not too many side effects, and then there always are side effects, so then you try to steer clear of those things. We're in a totally new age at this point, though, because a lot of drugs coming up are not just small molecules. So this, the Amgen pipeline has this key at the top, and they show you that some of the, there's different classes of these therapeutics. Some of them are antibodies, some are cancer immunotherapies, some are small molecules, some are protein, like peptide type of molecules. So traditional drugs were more typically small molecules. And that's because the reason for getting something small is that it can be diffused through the body pretty well. The larger the molecule, the more difficult it is to diffuse into different areas of the body. So small molecules have the most pleiotropic, you know, action. They work, you know, they can get all sorts of different places. What came up as a big hit were these antibodies. So you see on this chart here, all of these antibodies. You see a lot of their drugs in the pipeline are antibodies. You know, there was a great set of lectures. When was this? I think about 2 years ago. BioMarin gave lectures at the science seminar. You guys have been to the science seminar? Yeah, so that's an important thing to participate in. And they had a whole series of lectures from BioMarin, and they took everybody through the drug discovery and drug development and then eventually marketing process. I was floored. It was so useful. And you know, and I know some people that have been students at Dominican that now work for biotech companies. And there was a woman in my cell biology, Guadalupe Sierra. She did the master's program and worked in BioMarin, and then now she's working with a scientist who's branched off from BioMarin into another company, but she's located right here locally. So there. You know, this is a career path, getting involved with these things. So the companies are always developing a therapy. That's kind of what they do. That's how they earn a living. They can't just study things because there isn't any money in studying things. The federal government is the one that gives money for studying things. How does the federal government do that? You guys know? How do they give money to research? You ever heard like the National Science Foundation, for example, NSF? They fund some things. The NSF they fund more sort of general science. They're not so health related. As soon as something's health related, it's the National Institutes of Health. So they're funneling billions of dollars into the U.S. economy every year to do research on medically related phenomena. So that's important. So anyway, that's the funding of the study. But then when it comes down to, you know, a product, then the companies have to make a product. So what's more important, the study or the product? Well, they're both important, right? It's nice, you know, it's important academically to understand certain diseases and syndromes, disorders, etc. Be able to, you know, have an appreciation for what molecules are involved, what cells are involved, which tissues, which organs are malfunctioning. You need to know all that stuff. But knowing things is not necessarily, it's not a business. It's not something that you can, you know, typically you get a job doing unless you decide to, you know, teach or do some research, which is often backed by the, if it's health related, by the National Institutes of Health. So this other group, the company group, the good thing about them is they also do some work to try to understand diseases, but then they develop products. And then in the process of that, they can employ many, many people. So they employ many more people than the NIH employs. Even, even at that level, at national level, the companies end up employing thousands of people. And there's some very strong biotech companies and now pharmaceutical companies in the Bay Area. So this is definitely an option. So antibodies. Why would an antibody be therapy? We're going to talk about we're going to talk mostly about blood cells today. But I thought it was easier to jump into this at the start than it was later. Because once we get into the blood cells, we'll be into that subject. So this is sort of an aside, but especially the hemopoiesis, where blood cells are developing, they're maturing, and there's new ones that are constantly being produced. If you add some of these treatments, you can affect what kind of blood cells are produced. You can change the population itself. So that's pretty powerful. Okay, so antibodies can be made against a particular molecule, right? So at a certain point when we talk about blood cell maturation, there are cytokines that are responsible for blood cell maturation. Some of the interleukins. Actually, in the previous class, I had mentioned erythropoietin. Remember that? And erythropoietin is a good one to know. I mean, that's the kind of molecule that you'd be most likely to be asked on a test of mine, on a MCAT exam, on a job interview. It's a very important molecule, and the reason is that erythropoietin controls the level of hemoglobin. Just one, it's a small peptide, it's a cytokine, and so it's a protein therapeutic. Like up here on the chart, it's over here on the right, a protein or peptide signal that has some receptors on the target cell, and it can trigger production. The cell will alter its behavior based on the fact that the cytokine is present. So erythropoietin will stimulate the production of red blood cells, but it's doing that because more hemoglobin is made. And so when you're budding off a bunch of red blood cells, having a lot of hemoglobin around is kind of a signal to say, yeah, we can make a lot more red blood cells. So, it's a very simple system. The red blood cell is kind of a bag of hemoglobin once it's finally matured. So then the antibodies, the thing about, we can touch on this again at some other point when you're more ready for it, but I just wanted to introduce this idea that that everything that we're talking about for modulating blood cell populations and behaviors, there's signals. And those signals become fertile ground for looking at things that change those signals and therefore are therapeutic in nature. And you can change it by adding the signal itself, which is usually a small peptide, or you could use an antibody, which would do what? Is it going to add more signal or take away signal or alter signal? What do you think? Also, where do antibodies come from? They come from blood cell types, right? So lymphocytes are producing antibodies. But these antibodies, these are made synthetically, and it's lab genetic engineering, and they make an antibody. So the antibody binds to an antigen, and by doing that, it takes it out of solution typically. So you can see how powerful that is. We have ways of adding the cytokine by just engineering it and producing the protein itself, or we can take it away. We can make an antibody against it and then pull it out from the body. So there's too much signal to make a certain thing and it's leading to imbalance. We can decrease those levels by using these antibody therapeutics. So you see that they're pretty popular. They're useful for a lot of different things. Okay, so I don't want to, spend time on this, but I think it's useful to just be aware of that. I'm just scrolling down here to show you that there's several more of these antibody reagents, and some of these are blood. Okay. Okay, where we left off the last time is, I want to get into the hemopoiesis thing, the chapter 13, but we weren't quite done with blood. I think what really worked well though last time was showing you guys some slides, and we were talking about how those slides could look different if they were taken from a patient that has a certain disease. So that was pretty interesting because it makes it pretty obvious that some of these methods are diagnostic, and they'll show you something about what's going on. Once you understand, you couple the academic understanding, all of this reading that you're doing in this book. The book's pretty dense, huh? You notice that? It's like each page has got a lot of stuff to know and some figures and stuff, and then you kind of wonder, do I need to know that cell type? Do I need to know what a megakaryocyte is? Yeah, probably. But do you need to know, some of the intermediates in lineage? Probably not necessarily. Am I going to ask you about a basophilic erythroblast? Probably not. But I need you to know that erythrocytes develop from an earlier progenitor cell, and have some knowledge about how that works. So it's not just a matter of memorizing a bunch of names, but understanding the relationships between the different things. And in that kind of information, that knowledge will stay with you a lot longer. Okay, so I wanted to go over the leukocytes, the different kind of leukocytes. So you remember from that other class, we were sort of differentiating the erythrocytes. What's another name for an erythrocyte? A red blood cell, right? Synonyms. A while ago, I was when we were first designing this course, I said to some of the assistants, I said, "What's the difference between a reticulocyte and an erythrocyte?" And nobody could answer. But it sounds like a blood cell type, right? A reticulocyte, and it is. But it's an immature erythrocyte. So, and there's kind of, interesting. I mean, it's a pretty subtle difference. Both of them have gotten kicked out the nuclei, but there's still a little bit of synthesis of hemoglobin in a reticulocyte. So that's just some kind of detail to know. Anyway, let's look at. A job candidate in the bio department that's giving a talk right now. Okay, I can't make it. We're here. So these are nice figures overall, just to kind of get really used to the idea that that there are these different blood cell types and what the relationship is, and basically it's no big deal. Once you memorize a couple of names and you know the relationships between the cells, you should know them probably for good. So remember, we were talking about neutrophils being a type of leukocyte. What's another name for a leukocyte? A white blood cell, right? There's red blood cells, white blood cells. So leukocytes are that whole class of white blood cells. And then there's various participants here And we talked about neutrophils being, very common. So when we looked at your blood smear, we would see red blood cells, and then you might see a larger blue stained cell, and that's going to be typically a neutrophil. And that's because, if you look at the population of that, it's 54 to 62%. These eosinophils are a small percentages, and so are the basophils. So the neutrophils are the ones that are much more common and kind of interesting for that reason. But these things all have their special roles to play. Basically, these are the category here, these spotted cells. What category do those make up versus the ones that are not spotted, these two? You guys read this stuff, so you're experts. Come on.
Granulocytes.
Yeah, well, they're granulocytes. Okay, so those are granulocytes. What about these guys? The ones that don't have spots? Agranulocytes. Right? It's simple stuff. And sometimes it's confusing when you read it, but when you see it and discussing it and looking at pictures and stuff, it's pretty easy to say, yeah, the thing with the granules, those spots are granules. So the cells with granules are granulocytes, and cyte means cell, so they're granule cells, right? Granulocytes. And then the ones that don't have it are agranulocytes. So that's, one of the nice things about histology as a study is it was extremely important to characterize these different things. I mean, once we had good microscopes and we started picking apart the body and there's all these different things happening, we started describing what we were seeing, and a lot of it was microscopic, observation, and then we started using chemical stains, and at a certain point the cells would stain one way, and then a little bit later they might stain a different way. So some of the names for the cells are even based on how they stain. Isn't that funny? I mean, it's like the horse leading the cart. I mean, the cart leading the horse, or something like that. It's like kind of backwards, but it's kind of like that. So they looked in the microscope and they saw things with granules, so those are granulocytes, and then the ones that didn't have granules, those are agranulocytes. So if we, let's see, we go to, oops, that's not it. Oops, sorry about that. It's going to reboot. Okay, so I'm basically just going to review the types of granulocytes and the types of agranulocytes, and talk about platelets and blood clotting. So why are there so many different types of granulocytes? What's in a granule? Why are there so many granules in there? There's like different chemicals, right? That's the granulocytes are the types of cells that are going to go after these invading infections, bacteria. There's also some small worms and stuff that can get into your body. I mean, there's all sorts of nasty stuff. So there's got to be some kind of defense. And so these cells are the main line of defense of our bodies. And it's not just defense within the blood, it's in the tissues as well. So that's kind of amazing. How does that happen? You guys have some explanation for that? Daniel, can you explain that?
Don't the granules in the granulocytes contain lysosomal chemicals?
Okay, what are the lysosomal chemicals?
What are the lysosomal chemicals?
No, what is that class of what is a lysosome? What's what?
It's an organelle that.
Okay, what is lysozyme versus a lysosome?
Oh, lysozyme is the enzyme that kills the debris.
Kills debris. Debris is debris living.
Eliminates the debris.
Makes debris. So you start out with something living and then you end up with something that's not living. Okay. Yeah. So that's one of the things that could be in there. Okay, so lysosomes they can contain lysozyme. Lysozyme is an enzyme that degrades bacterial cell walls. So isn't that amazing that your body has, is making chemicals to degrade foreign material specifically? That's pretty advanced, It's not an arbitrary thing. It's not like we haven't been living on the earth for a long time, right? I mean, humans have come from everything else, and there's been a constant battling between different types of organisms, and we have developed ways to defend ourselves. And a big part of that is having white blood cells. And the granulocytes are the ones that are armed. Inside these granules, they've got a bunch of different types of chemicals that will kill other things. So when you see those pictures of a granulocyte, think, this is weapons to be used by the body against other things. And it's a blood cell, but then, it actually ends up defending us against infections all over the body. So it goes from the blood into tissues. But then we were saying that the blood system is a closed system. And so red blood cells don't appear all over the body. If they did, you'd get like black and blue spots and stuff on you, which can happen actually if you have some internal, some bleeding of these vessels or that kind of thing. Okay, so we're interested in what are these defenses, and we have leukocytes, white blood cells that are armed. The granulocytes are the ones that have a bunch of chemicals in them, in those granules, to release the contents of granules, and that will actually kill bacteria. And one of those that we mentioned is lysozyme, which is an enzyme that will digest bacterial cell walls. Lysozyme can be contained in lysosomes, but a lysosome is sort of a more general term for digesting vacuole. But it's contained within these granules, and neutrophils have it. Okay, some of the other chemicals that are in there, we've mentioned some of these. One of these is myeloperoxidase, which I was basically calling bleach. It generates hypochlorite. Hypochlorite is bleach. If you look on a container of bleach, it will say hypochlorite. So that's what bleach is. So MPO is myeloperoxidase. That's one of the chemicals in these granules. There's a small peptide called defensins in there, and that can, it's a protein, but it can get itself into the cell membranes of bacteria and help to kill them. So that's what you should think of when you think of granulocytes. So there's a bunch of stuff in there. that can be useful as weapons against invading things. And then we were saying that, okay, the circulation is a closed system, and these things are armed, and they must get out of the circulation somehow. And so I think they do somehow get out of the circulation. So let's have somebody come up and explain to us that part. Any volunteers? You'll have a go at it? Okay.
Yeah. You want my drawings?
Yeah, you want to explain that? How does that work? So we have a closed blood system, and here's red if you want.
I don't know. So in general, it's a closed blood capillary, and you got your white blood cell guy comes along, and he needs to get into the interstitial space down here. So they actually sit here, and these guys kind of loosen up, and he kind of squeezes through the space in the capillary.
Okay. Yeah.
Right?
Yeah.
Then you can eat something up.
Yeah. So if he's chasing after a bacteria or whatever in tissue, he can actually get through the vessel. What kind of cells are in the vessel?
Epithelial cells. Or is it endothelial?
Endothelial. Yeah. There's different classes of cells. There's epithelial cells. Epithelia is the stuff on the outside. So on your skin, the outside of your skin is epithelia. But it's a general name. Epithelia is a general name for what's on the outside. When it's on skin, we call it epidermis. But if it's facing the outside, like the lining of the lung, it's a lung epithelia. But wait a second, the lung is inside the body. I thought I just said that epithelia are on the outside. Well, the inside of the lung, where it's facing air, that is the outside. Right? So, when you think of the organs of the body, the inside and the outside, the outside is facing either air or fluid, typically. And so that's always epithelia. So I think that the endothelial cells, so we Connor was just saying that, okay, these are endothelial cells that make up this vessel. So cells, they make up a tube. I mean, they're connected to each other, and they surround each other, and they make a big funnel. That's pretty cool. So this gets into the thing that we all want to talk about, which is how do cells form structures? And, structure function stuff. There's a lot of interesting biology there. We started the course talking about blood, and blood is about cells free in solution. So they're floating around in solution. They're kind of round, typically. That was one of the hallmarks we said of a blood cell, human blood cells. Everything tends to be round in solution because there's equal pressure from all sides. And so there's not much internal structure to make it a certain shape. There's no reason to do that often. So then, they tend to be round. Whereas an invading, a piece of yeast we looked at, the yeast looked about as big as the neutrophil. It was kind of this oblong shape thing. Well, that's, that elongated shape, that's not going to be something human. So, if you are presented with pictures of tissue, you'll be able to identify the possibility that, well, this is probably not a human cell. And then there's the neutrophil next to it. Well, what's happening? Well, the neutrophil is going after this thing. Okay, so so the neutrophil could be the thing in this vessel, and the neutrophil is going to make its way through this endothelial cell lining. So the actual tube itself is all endothelial cells. Yeah, there's some proteins that probably are made, but it's mostly cellular. It's very cellular. If you look at the tube, in cross section, it looks like a bunch of cells. This is the cross section of some sort of blood vessel or microcapillary, and you see all the cells around that, and that forms the tube. So it's actually, it's made up of cells. But because it's made out of cells, cells are individual units. They're like cobblestone. Sometimes they use that word cobblestone, and they're individual units. So therefore, if you wanted to squeeze between the unit there, you could do that, right? If you're, if these neutrophils are blue, just stain them blue. But if you wanted to squeeze through, it's not like you have to break anything, right? They're individual units, so you can kind of create a separation by moving them aside, and then there's a space where the cell can sweep through. And that's what Connor was drawing here, right? That was your rendition of that. Yeah. I'm just showing it in cross-section it makes sense because it's just individual blocks, individual cells, and they could be squeezed apart and you could squirt in there. How about red blood cells? Can a red blood cell, here's the red blood cell, so it's this disc. Can those squirt through? No. Why not? Connor, why can't they squeeze through?
They're just not mobile like that. They don't have the motility to actually the leukocytes have actual proteins on them that allow them to slow down, semi-adhere to the endothelial cells, and then work their way through. The red blood cells don't have those proteins, they just float on through.
Yeah. It's not their job, right? There's no reason to have the red blood cell or erythrocyte to have their erythrocyte in the interstitial fluid of the body. And so, I addressed that a lecture or two ago, and I said, well, what if you see a red blood cell in a tissue slice? What does that mean? And then I said, well, that's probably an artifact, right? It doesn't belong there. If there was a damage, there could have been some bleeding. If you've sliced into an area which was black and blue, that's going to have some drying up, red blood cells, and so you're going to get that effect. But generally, it's sort of an artifact. Anything that you biopsy and slice or whatever, you're going to damage some vessels, and so you are going to see some red blood cells. But in general, that's just an artifact. It's due to the preparation. And so in actual in vivo situation, it's not, you don't have red blood cells, but you do have these neutrophils and other granulocytes and agranulocytes as well, but they have an amoeboid movement. They're able to actually squirt through. It's their job to get across that in between the cells. And I drew it in cross section here, showing you their individual cells, and you just sort of move the two cells apart and you can kind of squish in there. You don't have to destroy anything. You don't have to break anything. You don't have to eat your way through. You just need to push aside the surrounding cells, and then you're in. And so then once you're in the interstitial space, you can chase after a bacteria or a yeast or some sort of invading organism. And you're going to do that using what you're armed with. And these granulocytes have granules. And granules are bags, little bags of chemicals that the cell can throw at the invading organism. One of these is the ability to make hypochlorite, which is bleach. And if you guys worked in a lab, have you seen, like, a lot of times we decontaminate everything with bleach. I'm constantly using bleach. I use gallons of bleach in my lab. And so, when I aspirate using a Pasteur pipette and I'm taking some medium off, it goes through a tube and it goes into a vacuum flask. And at the bottom of the vacuum flask, I've got a bunch of bleach. And I do that because if there's any kind of microbe or whatever in there, and the vacuum flask itself is not sterile, so stuff could start to grow. But with bleach present, not that much is going to grow. It's definitely going to inhibit the growth of all sorts of things, including yeast, bacteria, and molds too, to some extent. Depends on the final concentration, but it's definitely a powerful bactericidal and other cytal activities. So it has that, and we also mentioned Lysozyme and Defensins. defensins are a class of small peptides that can interfere with the membranes of bacteria, and they also seem to have some other activities that we don't quite understand completely yet. But it's an interesting class, and there's many different types of defensins. And in other parts of the body, they're called different things. Like in the gut, it's a cryptin. So cryptins are just a class of, just another variation of the alpha defensins. They probably have specific, arch enemies that they're going after. So the structures are been altered a little bit. Okay, so so we just wanted to review the neutrophil. Why does this strain people? Any suggestions what we should do? Yeah, I was picking on Michael all the time for this, but this seems particularly buggy. I don't know why. What would you escape on it? What? Okay, yeah. So this was a picture of neutrophil in cross-section, and it shows the what they call lobulated nuclei. Seems like there's more than one nucleus. So the reason why we're talking so much about the neutrophil is because most of the leukocytes, white blood cells, are neutrophils, and they're the guys that are often squirting through the capillaries into interstitial fluid, chasing after our arch enemies, bacteria or yeast or other sorts of infection. And they have this nuclei which is lobulated, sort of in different lobes to it. And one of the thoughts there is that, well, it's kind of a floppy nucleus. If you had a big round nucleus, it's harder to squirt through things. But if you it's all connected, but if you make it kind of different domains, it can kind of go in different directions, so it's more flexible. So this is a sign of a flexible cell, that's what they think. And then we were talking about the different sorts of granulocytes that are in there. There's specific granulocytes as well, which can have different sorts of chemistries. But anyway, a picture like this, you would be able to identify what that is. Then this is a eosinophil, and you see here's a difference between a neutrophil and an eosinophil. They're similar. They're both about the same size. They're both granulocytes, so they're going to have granules. The usefulness of those granules changes a little bit. Eosinophils have this major basic protein, NBE, and they have peroxidase and toxins. They're good for killing things like worms, which is kind of interesting.